Hello, everyone. Thank you for attending today's webinar titled LakePharma's Coronavirus Spike Protein Antibodies, a fresh quiver of arrows against the crowned invader. My name is Matt Harrington. I'm Senior Manager of Operations at LakePharma, and I'll be today's moderator. I am thrilled to host today's webinar. Before we get started, please feel free to submit any questions to our speaker at any time by clicking on the Q&A button located on the toolbar at the bottom of the window. If we cannot get to your question today, we will respond via email. And now, I would like to introduce today's speaker. Dr. Brian Zabel received Bachelor of Science degrees in Biology and Mathematics from MIT in 1997 and a Ph.D. in Immunology from Stanford University in 2004. Dr. Zabel holds 9 U.S. patents and has published over 50 peer-reviewed research articles in high-impact journals such as the Journal of Experimental Medicine. Dr. Zabel has special expertise in hybridoma-based monoclonal antibody discovery with demonstrated success against challenging targets. At LakePharma, Dr. Zabel leads a scientific team of hybridoma experts in the discovery of therapeutic and diagnostic antibodies. And now, we turn over to Dr. Zabel.
Thanks, Matt. Right, so today we're going to talk about our coronavirus spike protein antibodies, a fresh quiver of arrows against the crowned invader. I just wanted to show this photo here. So this is what LakePharma 201, this is our San Carlos site, which is where we are located. This is how it looks most days. So like today, nice blue skies. But on September 9th, we were in the midst of the wildfires and it was just a wild day and the skies turned orange and this is what it looked like outside our building. So, I mean, 2020 has just been a wild year. So anyway. So if you're interested in antibody discovery, start with hybridoma.com. So this is a website that we put together. It contains a lot of good educational information on hybridoma-based antibody discovery. And it also covers our antibody discovery workflow, campaign success, some of the innovations that we've got going here at LakePharma. Our group is called Discovery Immunology, and we operate at the end here. Near target discovery, we work on antibody lead discovery, screening, and lead characterization. LakePharma is a contract research, development, and manufacturing organization. So we work on this end of the arrow or in the chevron diagram. And Lake can cover everything all the way to GMP productions for material going into clinical trials. So recently, I'm proud to announce that we achieved ALAC accreditation. So ALAC is the Association for the Assessment and Accreditation of Laboratory Animal Care. We became fully accredited in June of 2020, so it's a lengthy process. It requires lots of oversight, and so our vivarium and animal program is fully accredited, and we perform our own mouse and rat immunizations, and we do all of the in vivo and hybridoma-based antibody discovery work at Lake. I just want to point out the excellent sequence diversity that we've discovered in probably over 100 campaigns here at Lake. In this dendrogram here, there are probably 20% of the sequences that we've identified. This is an alignment that was put together using a muscle alignment with neighbor joining, using Genius software. Campaigns are color-coded, so you can see for some campaigns there are clusters of sequence-similar antibodies, and in other campaigns, like in green here, in other campaigns, the sequences are sort of randomly distributed. I think, for comparison purposes, we included cetuximab, rituximab, and infliximab. These are just unrelated therapeutic antibodies. Variability in CDR3 lengths within and between antibody discovery campaigns is a way to look at antibody diversity, and these are different campaigns here, and this is just looking at CDR3 lengths. And this slide is just to point out, some of the diversity that we get in our hybridoma discovery campaigns. Another way you can assess diversity is by looking at the V gene usage in the heavy and light chains. And so far, we've seen a large number of distinct VH and VL genes represented in the sequences that we've discovered. And the frequencies can range from 0.3 to 10% for any given allele. So it's a very productive and successful approach for antibody discovery. And so now getting into more of the topic today, the world really changed in Q1 of 2020 with COVID-19 and with SARS-CoV-2. So in Q1, the genome sequence of SARS-CoV-2 became available, and Lake immediately started producing recombinant viral proteins for use in diagnostic tests. Our CEO, Hua Tu, he's previously presented a couple of webinars on these proteins, and they're being used in diagnostic kits across the country. And around that time, we also joined about 200 other companies and academic groups in vaccine development. And as a contract R&D and manufacturing company, we have all the resources, but this is really our first foray into drug development, hence sort of the big pharma versus Lake Pharma depicted here. So our first generation COVID-19 vaccine is LP151. So we really focused on the RBD domain here of the spike protein for our vaccine. So the spike protein attaches to the host cells through the receptor binding domain interactions with the angiotensin converting enzyme 2 here, the ACE2. And it's been shown that neutralizing antibodies against RBD prevent viral infection of ACE2 positive cells. And we ended up testing LP438, which is the RBD domain, with a series of different adjuvants in our immunologically diverse Penta mice for anti-RBD plasma titer and for spike ACE2 binding neutralization. We identified a couple of coadjuvants, coadjuvant 218 and 315, as superior in inducing persistent neutralizing titers in all 5 of the Penta mice strains that we use. I did a previous webinar on Penta mice, and we can talk about that, but they're very immunologically diverse. So altogether then, LP438, which is the RBD, plus these coadjuvants form LP151, which is our first generation lead vaccine. And what we have ongoing are safety and efficacy studies to support an IND application to the FDA, which is planned for November. And if all things go well, we could be in phase 1 clinical trials anticipated in January of 2020. So like I mentioned, we use Penta mice to evaluate our vaccine for immunogenicity, and we think it's an ideal platform for doing so. So it's a proprietary immunization platform. It comprises 5 unique mouse strains that we generate through in-house breeding, covering 9 different MHC class 2 haplotypes. So we have 10 mice per vaccination cohort, and this really provides maximum immunologic diversity. So the animals are shown here, and their strains are listed here. Their names are based on their MHC class 2 haplotypes. So we believe that the Penta mouse platform really is a better mimic of the immunologic diversity that's present in outbred human patients, as opposed to a single inbred strain like a B6 mouse. So in identifying an optimal adjuvant for LP438, we looked at a panel of adjuvants and assessed activity by ACE2 binding inhibition with the spike protein, and that's what's measured on the y-axis here. So here we're looking at ACE2 inhibition. Basically, it's IC50, so at different plasma dilutions here. So it's a titer measurement. And you can see for comparison, we have CFA. So CFA is a very strong irritant, very good adjuvant. We actually add a couple of other things in with it. So this is sort of like the maximum response that we predict you could get. All the animals in the Penta mice strains here are color-coded for comparison purposes. And so this is sort of best case scenario, I would say, with CFA. And then these are the different adjuvants that we looked at. And you can see there's definitely some interesting strain-dependent responses. So with certain adjuvants, you can get really good titers, and in other strains, that adjuvant combination doesn't work well. So we selected a combination of adjuvants that gave a very strong titer response in all of the strains of the Penta mice, with the thought behind that being that this is sort of a best case scenario that would result in efficacy in the largest outbred human patient population, or human population. So, so this is where LP151 came from, and I mentioned that because in parallel with our vaccine work, we started doing parallel antibody discovery work to try to discover antibodies against the full spike protein, the spike trimer protein of SARS-CoV-2. And as I mentioned, Hua has already presented quite a bit of information on the spike trimer. Down below here, we have some QC data from the LakePharma produced recombinant spike protein. And you can see here that we get good ACE2 binding, which is a great QC step. You know, it should be, if it's properly folded, it should bind to human ACE2. And also, we've got plasma samples from COVID-19 convalescent patients, and those plasma samples also recognize the recombinant protein that we produce here. So it's a good antigen, and we used it to immunize mice. And we're focusing here on 4 animals from our Penta mouse cohort. And this is looking at the titer, spike protein titer on day 12 and on day 19. And we got really robust and rapidly developed titer in these animals. By day 19, which is pretty early for us, they already reached our titer threshold, which is giving a greater than 100,000 RLU at a 1 to 50,000 plasma dilution. So we move these animals for a final boost and fusion. In addition, we also analyzed the plasma from these animals, and we found very nice neutralizing immunoglobulin responses in those day 19 animals here. So this is these are the IC50. So it's a neutralization titer experiment. So at 1 to 3,000 dilution of plasma, we can get very nice inhibition of ACE2, of spike protein binding to ACE2. And not only do we have that measure, but we also have infection inhibition data using a SARS-CoV-2 pseudovirus infection model. So these studies were performed in Li Jun Rong's lab at the University of Chicago. Basically, it's an HIV backbone that can't replicate on its own, but it can infect. And in these constructs, you can overexpress different virus spike proteins, and they overexpress the SARS-CoV-2 spike. And we're able to show here, so plasma 3 is from our vaccinated, immunized animals, and you can get 100% neutralization of infection of the pseudovirus into ACE2 positive TMPRSS2 positive cells at a 1 to 100 dilution here. And we also see some SARS-1 pseudovirus infection inhibition as well, and nice titers here. So good antibodies were being produced by the animals, neutralizing antibodies were produced by the animals. So all signs pointed positive for moving into antibody recovery. So we performed electrofusion and plated out the hybridomas into 10 384 well plates. We ended up screening about 10,000 hybridomas by ELISA for reactivity against the SARS-CoV-2 spike protein. And we used a His control as a counter screen. The spike protein that we produced had a His tag. And so we identified lots of spike protein specific hits with high ELISA signals that were selective against His. So this is, 2,900 different wells that are giving very nice ELISA RLU, greater than 100,000. And we also did identify some wells that had His antibodies that are potentially interesting down the road. So we went ahead and picked our top 375 SARS-2 spike binding hits and our top 9 His binders and moved them forward. So into downstream screens. So in our secondary and tertiary screens, we're interested in identifying antibodies as they bound to the RBD domain of the spike protein. And that's shown in orange here. And then we also, in our first pass, wanted to look at cross-reactivity to 2 other coronavirus spike proteins, the MERS and the human coronavirus spike protein. So interestingly, over half of the antibodies from the animals immunized with the entire spike trimer, half of them bound to the RBD domain, which is interesting. The RBD domain is only about 15% of the total spike protein, so it seems to be very antigenic. And in addition, on the left-hand side here, these are all antibodies that are cross-reactive against the other 2 coronaviruses that we looked at, the MERS and the HKU1. Over on the right-hand side here, these are antibodies that are very selective, on the other hand, for just the SARS-CoV-2 spike, and they're non-RBD binders. And now in this one, we swapped out RBD for SARS-1, and this is another coronavirus, and approximately 2/3 of the antibodies actually cross-react and bind to the SARS-1 spike protein, and that's shown in orange here. One of the follow-up experiments we did in the tertiary screen was to assess neutralization, so the ability of our hybridoma soups to block the binding of the spike 2 to ACE2 in that biofunction ELISA that I mentioned earlier. We found 70 that were really nice blockers here. We had some intermediate blockers, and then we had some weak or non-neutralizing antibodies in this biofunction assay. And we also had the soups analyzed for pseudovirus infection inhibition as well. And we got a nice collection here of antibody soups that inhibited greater than 90% pseudovirus infection. So these are real functional inhibitors here. In this group, these are partial inhibitors. And in this group, we identified a single hybridoma well, 10b13, it's kind of blown up over here, that also was able to inhibit SARS-1 infection of target cells. So I think it's a special antibody, 1 out of 375 that's able to inhibit both SARS-1 and SARS-CoV-2 infection. And then we have a large group here of SARS-CoV-2 binders that are non-neutralizers. And I think it's interesting to point out that, most of the antibodies that we were able to recover from the animals that were immunized with the spike trimer were actually non-neutralizing antibodies. And of those non-neutralizing antibodies, most of them were non-RBD binding antibodies. Whereas if you look at the neutralizing antibodies here, the top like 30%, most of these are actually RBD binders. So I think we're using the hybridoma information to inform a bit on the vaccine work. So if the goal of the vaccine is to get neutralizing antibodies, I think going after the RBD domain would be favorable. Clearly you get a lot of non-neutralizing antibodies when you immunize with just the full spike trimer, and some of those might be problematic in terms of leading to antibody-dependent enhancement of disease or the potential for that sort of thing down the road. So we looked at our data, we lined it up. This is what we call a master file. We line up all of the binding or activity profiles, functional profiles here, and within each column, they're color-coded based on the intensity of the signal or the response in the column. Over on this hand, and everything is sorted based on the pseudovirus infection inhibition, looking at SARS-CoV-2. So these were the top sort of neutralizing antibodies here, and then looking across the board at different reactivity profiles, cross-reactive against the other coronaviruses. Do they bind to S1 domain, S2 domain of the spike protein? Here's that 10b13 that inhibited SARS-1 as well. So we selected a diverse panel of hybridomas, which is about 10% of our total leads, for single-cell cloning, sequencing, scale-up, and purifications. And the sequences that came out, again, were pretty diverse. So this is a dendrogram display of our spike protein antibody sequence relatedness. Again, generated in a similar way where we had concatenated the VH and BL sequences and then used software to produce the alignment chart here. And again, we include rituximab, infliximab, and cetuximab, unrelated antibodies, just for comparison purposes here to illustrate sort of the sequence relatedness. So a diverse panel here, and again, by looking at pH, gene usage in the heavy chain and in the light chain, there's quite a few different alleles of both the heavy chain V gene and the light chain V gene that are used. Distinct alleles were used in 75% of the discovered antibodies. And in most of the antibodies, the VL-CDR3 is 9 residues, which is in orange here, which is the most common length of light chain V gene CDR3s. And the heavy chain, there's quite a bit of range in size there. They go from 7 to 18 residues in the CDR3 of the heavy chain. So good diversity. And we looked at, we took the purified antibodies and looked at a battery of different assays to actually put some numbers on some of these binding profiles. So we're looking at basically concentration-dependent binding and functional activities of the purified antibodies by ELISA. So we're getting potency values here against the full spike trimer of SARS-CoV-2, just the S2 domain, just the S1 domain, the RBD domain. The RBD domain is within the S1 domain. So these two binding profiles look very similar across these different antibodies here. We did include a couple of control antibodies from cynobiologicals as well. These antibodies, these antibodies here are ones that cross-react and also bind to the SARS-1 spike protein. We have one antibody that binds pretty nicely to the MERS spike protein, some lower potency binders here as well. Same thing for HKU1. And then we also assessed them in that pseudovirus infection inhibition assay, and we found some very nice potent inhibitors there in that assay as well. And they also block spike protein binding to the ACE2. So in this table, this is just a numerical table now of the potency, affinity, and selectivity of our selected SARS-CoV-2 spike binding antibodies. We also have the isotype information in this column. So we've got a variety of IgG1s and IgG2Bs. Looking at this guy here, so 10B13A, this was, again, the only one of our hits that inhibited both SARS-CoV-1 and SARS-1 and SARS-CoV-2. And the inhibition potency is shown here. So it's a 800 picomolar potency in terms of SARS-CoV-2 inhibition, and 5 nanomolar for SARS-1. The binding affinities were determined by Cartera, and they're shown over here. So single-digit binders here in terms of RBD, which is the antigen. This guy is 14.9. The cynobiologicals antibody has a relatively lower affinity binding at 35 nanomolar. Also pointing out that 10O24A is probably our most potent pseudovirus binding inhibitor here, 340 picomolar potency. So I think there are some good therapeutic candidates available in this list, and there's also some interesting diagnostic profiles here if interested in sort of being able to specifically identify SARS-CoV-2 versus other coronaviruses, or maybe it's of interest to identify multiple coronaviruses, and then as a percentage of that, you could use the more selective ones. So it's a pretty well classified and characterized list of antibodies. So overall, in summary, we've gone over a lot of this, but we use the Penta platform, which we immunized with SARS-CoV-2 spike trimer protein, identified 350 hits. All of these candidate parental hybridomas have been cryopreserved. 33 of those we selected for monoclonal hybridoma generation, 100 mil scale-ups and sequencing. We have good sequence diversity, which has been obtained. We have good potency and affinity values and selectivity for 10 so far of our SARS-CoV-2 binding antibodies. Again, this guy 10B13A is an interesting one, potential therapeutic candidate, as well as some of the other high potency SARS-CoV-2 binders. We've got some S1 and RBD selective antibodies, some S2 selective antibodies with different cross-reactivity profiles. And pretty much all of these antibodies are available for licensing for therapeutic and diagnostic applications. So if you're interested, please send me an email and we can set up a call to go over what you're interested in. So I'd like to thank everybody who's helped out on this project in Discovery Immunology. Xiaomei, Dan, and Jeff have been our preclinical COVID-19 task force, doing most of the R&D work in the group. Ling helped out with the antibody diversity analysis. Natalie's our vivarium specialist, helping out with the pentamyce breeding. and Carrie Ankush and Vaidehi are our scientific managers that keep all of our projects on track and within good time frames. In antibody engineering, I'd like to thank Margaret and Lachun, who've done the affinity analysis to give us our KD binding numbers for the RBD binding antibodies. And again, if you're interested in the spike antibodies or in SARS-CoV-2 proteins or in hybridoma-based antibody discovery in general, please give me an email or go to our website, hybridoma.com or LakePharma.com. There's more information available on the web. And this is our team here in Discovery Immunology, being safe at work, with the fully masked and social distancing, staggered shifts, all of that fun stuff that everyone's been dealing with over the last 7 months or so. So thank you very much. And we'll move into question and answer.
Thank you, Dr. Zabel, for your presentation. Now we would like to open up the floor for questions. A quick reminder that you can submit questions by clicking the Q&A button located on the toolbar at the bottom of the window. Dr. Zabel, can you expand more on the advantages of the Penta mice platform?
Sure. So let's see. I don't know if it was 2 or 3 months ago, we did a webinar on Penta mice where we kind of go into how we generate them and what, some of the functional profiling and what their MHC diversity looks like. So that's available on hybridoma.com. But in general, I think what the main thing that the Penta mice platform provides is diversity. So enhanced opportunities for presentation of peptides on MHC that, maybe a B6 animal's MHC can't present a peptide, whereas one of the members of the Penta mouse platform is able to present that peptide. We know in vivo you need to have a good T-cell help to provide strong antibody responses and good affinity maturation in vivo. And so by casting a wide net, by using a diverse set of mice at the outset, which is what we provide in our proprietary but royalty-free Penta mice platform, basically gives you multiple chances in one go of getting a strain or 2 or 3 or maybe, who knows, but it gives you a better chance of getting those high affinity antibodies that would otherwise be limited if you're just using a single inbred strain like a B6 or a BALB/c mouse. So I think it's for vaccine development, it's an interesting platform because it more closely models outbred humans, which have a lot of immunologic diversity as opposed to just a single B6. So the MHC in the Penta mice platform is 40 times more diverse than what you'd have in a B6 mouse.
How many animals were used to generate the 10,000 hybridomas?
That's a good question. So we just used 4 animals, but the lymph nodes and the spleens were very large, and we have a lot of cryopreserved material. So we only used a fraction of the material from those animals that we moved into fusion for the discovery of those initial set of antibodies. And we played out 10 384 well plates, and our fusion efficiency was, I think, between 2 and 3 hybridomas per well, which leads to the calculation that we screened 10,000 hybridomas. So there's plenty of material left in the freezer if we want to go back and screen or look for very specific funneling or types of antibodies.
Does your RBD neutralizing antibody completely avoid ADE effect?
Yeah, we don't know that. So ADE, as far as I know, there aren't any in vitro ways to predict that clearly. So I feel like ADE is something that you have to assess in vivo. There isn't a great mouse model to assess ADE. And hamsters actually don't, as far as I know, even though they can be infected by SARS-CoV-2, they don't model that aspect of it. You almost need to go into a non-human primate model to evaluate ADE. And I haven't seen, there's so much literature coming out on COVID-19, but I haven't seen a model of that yet. So I think that's something that would have to be tested either actually clinically or in an NHP study.
Can you explain the difference in IC50 between the pseudovirus and spike ACE2 assays?
Yeah, I mean, there are different assays. The pseudovirus infection inhibition is more closely, I think, relevant to what you would be, experiencing in vivo. It's using, it's a cell-based assay. The pseudovirus has a luciferase. And so I think the potency there is probably more in line of, what you would see. I guess the next step up from that would actually being the SARS-CoV-2 infection plaque assay, which would be done in a BSL-3 lab. We use the IC50 in the neutralization assay, the spike and the ACE2 binding as a guide, and they're pretty they correlate. So we've done some correlation analysis, but you will identify some antibodies that will block better in the pseudovirus than and have better or more higher potency in the pseudovirus infection inhibition than what you see in the biofunction ELISA. So I think, we might be missing some antibodies there or some part of the process by, if you rely too much on the spike ACE2 binding. So spike ACE2 is a good guide. And then I think pseudovirus is higher, strength assay in terms of clinical relevance. And then you've got the next level up, which is the actual virus infection.
Can you focus your responses to a particular epitope through the immunization and screening protocols?
Yes, that is definitely possible. We've seen that with doing work with RBD versus the full spike protein. So, for example, in the RBD is just a small, 10 to 15% domain of the full spike. So you can definitely focus on domains, and you can do that with S1 and S2 as well. The smaller the peptide, basically you want your whatever you're immunizing with to pass a QC. So it's got to be folded properly, and as long as it either binds a reference antibody that is works, or binds a ligand that is critical, it embodies the ACE2 binding, for example. If you're talking about RBD, then you can move forward with confidence.
Excellent. Thank you, Dr. Zabel, and thank you everyone for attending. Again, if you do have any further questions for Dr. Zabel, you can email him at brian.zabel@lakepharma.com. Thank you all for attending today's webinar.